Gene families and evolution of gene clusters. Most recent common ancestor of all living things, bacteria, eukarya, and archaea. When we just enlarge that eukarya portion, we could see chimpanzee, human, and mouse like this. Then comparing distantly related species. Highly conserved genes have changed very little over time. This helped clarify relationships among species that diverged from each other long ago. Bacteria, archaea, and eukaryotes diverged from each other between 2 and 4 million years ago. Highly conserved genes can be studied in one model organism and the results applied to other organisms. So, one more thing, highly conserved genes have changed very little over time. This helped clarify relationships among species that diverged from each other long ago. Bacteria, archaea, and eukaryotes diverged from each other between 2 and 4 billion years ago. Highly conserved genes can be studied in one model organism and the results applied to other organisms. Human and chimpanzee genomes differ only by 1.2% at single base pairs and by 2.7% because of insertions and deletions. Several genes are evolved faster in humans than chimpanzees. These include genes involved in defense against malaria and tuberculosis and in regulation of brain size and genes that code for transcription factors. Then humans and chimpanzees differ in the expression of the FOXP2 gene, whose product turns on genes involved in vocalization. Differences in FOXP2 gene may explain why humans but not chimpanzees communicate by speech. Comparing genome sequences providing clues to evolution and development. Genome sequencing and data collections has advanced rapidly in the last 25 years. Comparative studies of genomes advance our understanding of the evolutionary history of life, help explain how the evolution of development leads to morphological diversity. Help explain how the evolution of development leads to morphological diversity. Then comparing genomes. Genome comparisons of closely related species help us understand recent evolutionary events. Genome comparisons of distantly related species help us understand ancient evolutionary events. Relationship among species can be expressed by a tree-shaped diagram called a phylogram. Gene family is a set of several similar genes formed by duplications of a single original gene and generally with similar biochemical functions. A gene family is a set of homologous genes within one organism. When a gene is present in two or more copies per genome, the condition is known as redundancy. Two or more copies. When a gene is present in two or more copies per genome, this condition is known as redundancy. The numbers of a gene family may be either clustered together, dispersed on different chromosomes, or present in a combination of both. Gene cluster is a part of a gene family. A gene cluster is a group of two or more genes found within an organism's DNA that encode for similar polypeptides or proteins. A gene cluster is a group of two or more genes found within an organism's DNA that encode for similar polypeptides or proteins which collectively share a generalized function and are located within a few thousand base pairs for each. Once again, a gene cluster is a group of two or more genes found within an organism's DNA that encode for similar polypeptides or proteins which collectively share a generalized function and are located within a few thousand base pairs of each other. So, the size of gene clusters can vary significantly from a few genes to several hundred genes. Genes found in a gene cluster may be observed near one another on the same chromosome or on different but homologous chromosomes. Extensive tandem repetitions of gene normally occur when the gene product is needed in unusually large amounts, for example, genes for rRNA, histone genes, etc. Sometimes all the members of a gene family are functional, but often some members are non-functional, for example, pseudogenes. Genes are multi-gene families. Many eukaryotic genes are present in one copy per haploid set of chromosomes. The rest of the genes occur in multi-gene families, collections of identical or very similar genes. Some multi-gene families consist of identical DNA sequences, usually clustered randomly, such as those that code for rRNA products. Evolution of gene superfamilies. Large set of genes divisible into smaller sets of families. Genes in each family more closely related to each other than to other members of the family, arise by duplication and divergence. If the genes of a gene family encode proteins, the term protein family is often used in an analogous manner to gene family. An example for such family are the genes of human hemoglobin subunits. Protein family. Model for evolution of vertebrate globin genes. Time tree. Look at this figure carefully. If you need to read all this, just pause the video. An evolution of genes with novel functions. The copies of some duplicated genes have diverged so much in evolution that the functions of their encoded proteins are now very different. For example, the lysozyme gene was duplicated and evolved into the gene that encodes alpha-lactalbumin in mammals. Lysozyme is an enzyme that helps protect animals against bacterial infection. Alpha-lactalbumin is a non-enzymatic protein that plays a role in milk production in mammals. So, a great amount of diversity is there. So, duplicated genes have diverged so much in evolution that the functions of their encoded proteins are now very different. Developmentally controlled complex multigene family, human globin gene cluster, products, organization and evolution. Products of hemoglobin genes. The hemoglobin molecule is a tetramer and is composed of two similar polypeptides, the alpha and beta chains encoded by two distinct genes. Each polypeptide incorporates a heme group that reversibly binds oxygen. Heme group. Each polypeptide incorporates a heme group which irreversibly binds oxygen. So hemoglobin, heme protoporphyrin plus Fe2 plus globin alpha and beta chain. Later when we zoom this image, we could see red blood cell, beta chain, alpha chain, iron, heme group, beta chain, alpha chain, helical shape of the polypeptide molecule. Helical shape. So hemoglobin, heme protoporphyrin plus Fe2 plus globin alpha and beta types. The globin genes are coordinately turned on and turned off during the embryonic, fetal and adult stages of development. The genes for alpha globin lie in a cluster on chromosome 16, while those of beta globin are located in chromosome 11. Chromosome 16 and chromosome 11. Now if I get these numbers, chromosome 16 and now chromosome 11. Globins, alpha clusters and beta clusters. 16 chromosome telomeric end near the telomeric end. Second one, beta cluster near the telomeric end, alpha cluster telomeric end. Various globin genes expressed during the embryonic, fetal and adult stages of development. Various globin genes expressed during the embryonic, fetal and adult stages of development. Alpha globins. Zeta genes are expressed during the embryonic development. Zeta genes are expressed during the embryonic development in case of alpha globins. Alpha 2, alpha 1 genes are expressed during the fetal and the adult stages of development. Zeta genes are expressed during the embryonic development. After that, alpha 2 and alpha 1 genes are expressed during the fetal and adult stage. Alpha 2 in the fetal stage, alpha 1 in the adult stage. Okay. Zeta in case of embryonic development. Beta globins, just remember where this alpha is situated, chromosome 16. Okay. So beta globins, epsilon genes are expressed. Earlier we learned about zeta genes. Now it's about epsilon genes. Epsilon genes are expressed during the embryonic period. G, gamma, A, gamma genes are expressed during the fetal development. Delta and beta genes are expressed during the adult stages of development. Delta and beta during adult stage. Delta, beta during adult stage. G gamma and A gamma genes are expressed during the fetal development. Globin genes and its products. Alpha globin, beta globin, heme alpha globin. So percentage of total globin synthesis post-conceptual age weeks, postnatal age weeks. So megaloblast, macroblast, site of erythropoiesis, normoblast, bone marrow, etc. can be seen. Beta globin locus chromosome 11, alpha globin locus chromosome 16. Embryonic, fetal and adult type. Human globin genes organization and expression, 3 exons. 3 exons. 3 exons coding type, 2 introns non-coding, and 3' and 5' UTR. 3 exons coding portion. 3 exons coding portion, 2 introns non-coding portion, IVS1, IVS2. Then 3' and 5' UTR. Promoter, enhancer and silencer. Promoter, enhancer and silencer portions. LCR is an enhancer, chromosome 11. Then beta globin gene cluster. Spacer DNA is non-coding DNA between gene family members. Pseudogenes are family members that do not code for functional mRNAs or proteins. Transcription factors for globin synthesis. General AP1, SP1, YY1, USF, SCL, TAL1. Then specific LCR F1, EKLF, ATXR, GATA1, F4G, NFE2, NRF1 and NRF2. AP1 and SP1 in general, TFs, transcription factors for globin synthesis, AP1 and SP1. Here specific NRF1 and NRF2. Regulatory regions in globin genes. Basal promoter, upstream regulators, proximal enhancers in 3' end of beta and gamma globin genes. Distal enhancers or master regulators of alpha clusters and beta clusters. Errors in basal or proximal regulatory regions result in thalassemia. Then basal promoters in beta globin gene. Sequence BRE, TATA, INR, MTE and DPE. RNA polymerase 2. Mutation in promoter. Stop expression and beta thalassemia. Upstream regulators in beta globin genes. Enhancers. Proximal 3' end enhancers in beta and gamma globin genes. Distal master regulators LCR and HS. Proximal enhancers at 3' end of beta and gamma globin genes. HBB, HBG1. Mutation in enhancers, down expression, beta thalassemia. Evolution of globin genes. Genes encoding the various globin proteins evolved from one common ancestor globin gene which duplicated and diverged about 450 to 500 million years ago. After the duplication events, differences between the genes in globin family arose from the accumulation of mutations. Subsequent duplications of these genes and random mutations gave rise to the present globin gene which code for oxygen binding protein. Subsequent duplications of these genes and random mutations give rise to the present globin gene which code for oxygen binding proteins. The similarity in the amino acid sequence of the various globin proteins support this model of gene duplication and mutation. Subsequent duplications of these genes and random mutations gave rise to the present globin genes which code for oxygen binding proteins. The similarity in the amino acid sequences of the various globin proteins supports this model of gene duplication and mutation. So ancient globin gene, hemoglobin gene, myoglobin gene, plant globin genes. So representation of evolution of ancestral globin genes. Ancestral globin gene insect like hemoglobin, then alpha subunit, beta subunit, hemoglobin, myoglobin. Then evolutionary time, duplication of ancestral gene, mutation in both copies, transposition to different chromosomes, further duplications and mutations. Ancestral globin gene is there, alpha globin gene family on chromosome 16 and beta globin gene family on chromosome 11. Duplication of entire gene can create multi-gene families, tandem gene family. Members of the multi-gene family are clustered on the same chromosome. Dispersed gene family. Members of the multi-gene family are on different chromosomes. Here you could see chromosome 1, chromosome 2, chromosome 3 and chromosome 3. They are just dispersed in time. So dispersed gene family. Unequal crossing over can expand and contract gene numbers in multi-gene families. Number of members in this cluster increase, number of members in this cluster decrease. Uncertain evolution can lead to gene homogeneity, unequal crossing over and gene conversion. Evolution of gene superfamilies. Large set of genes divisible into smaller sets or families. Large set of genes divisible into smaller sets or families. Genes in each family more closely related to each other than to other members of the family. Large set of genes divisible into smaller sets or families. Genes in each family more closely related to each other than to other members of the family, arise by duplication and divergence. Then evolution of globin superfamily. Secondary structure, tertiary structure, quaternary structure can be seen. Organization of globin gene families. Organization, alpha-like genes, active gene, pseudogene, ALU sequence, then chromosome, beta-like genes, chromosome, alpha-like genes, beta-like genes, etc. Then consensus splice sequences are there. 3' end cap site, then later 3' end cleavage signal, poly-A-T. Developmental variation in gene expression, alpha-like genes, beta-like genes, embryonic, fetal and adult human. Gene expression controlled by location, alpha, gamma, beta and delta. Epsilon, embryonic, yolk sac. Yolk sac, liver, spleen, bone marrow. Evolution of mouse globin. Evolution of mouse globin. The hemoglobinopathies, thalassemias. Anemias associated with impaired synthesis of HP subunits. Thalassemia can arise from different mutations causing a disease of various severity. Alpha thalassemias, then beta thalassemias. Mutations of beta globin cluster are of different type, gene deletion. Mutations in beta globin cluster are of different types. Mutations in beta globin cluster are of different types. Gene deletion, transcriptional mutation, RNA processing mutations, RNA cleavage signal mutations, nonsense and frame shift mutations. Which are they? Gene deletion, transcriptional mutation, RNA processing mutations, RNA cleavage signal mutations, and nonsense and frame shift mutations. Non-coding regulatory region, exons, introns, intervening sequences, 3' cleavage mutant deletion, then RNA splicing mutant, transcription mutant, nonsense mutation, frame shift insertion and frame shift deletion. Main genetic mechanisms that contribute to the phenotype diversity of beta thalassemia. In Drosophila melanogaster, DNA genes are present in X and Y chromosomes. The cluster in X being much larger than that in Y chromosome. The region containing rRNA genes is often called rDNA, ribosomal DNA. Prokaryotes versus eukaryotes. Both prokaryotic and eukaryotic ribosomes can be broken down into two subunits. In prokaryotes, a small 30S ribosomal subunit contain the 5S ribosomal RNA. Copies of dispersed genome. This picture shows general organization of the repeating unit of rDNA, organization of the non-transcribed spacer in Xenopus laevis. Non-repetitive region 1 variable length, repetitious region 2 variable length, repetitious region 3 variable length. Next is about pseudogenes. A pseudogene is a segment of DNA that structurally resembles a gene but is not capable of coding a protein. Structurally resembles gene, it's a segment of DNA but is incapable of coding protein. Pseudogenes are most often derived from genes that have lost their protein coding ability due to accumulated mutations that have occurred over the course of evolution. Pseudogenes are ubiquitous and abundant in genomes. They were once called genomic fossils and treated as junk DNA several years. Nevertheless, it has been recognized that some pseudogenes play essential roles in gene regulation of their parent genes, and many pseudogenes are transcribed into RNA. Nevertheless, it has been recognized that some pseudogenes play essential roles in gene regulation of their parent genes, and many pseudogenes are transcribed into RNA. Pseudogene transcripts may also form small interfering RNA or decrease cellular miRNA concentration. The pseudogenes regulate tumor suppressors and oncogenes. Pseudogenes can arise through a variety of mechanisms. Spontaneous mutations in a coding gene that prevent either transcription or translation of the gene result in the formation of unitary pseudogene. Spontaneous mutations in a coding gene that prevent either transcription or translation of the gene result in the formation of unitary pseudogene. Duplicated pseudogenes are created via tandem duplication or uneven crossing over. These duplicated genes lose their protein coding potential due to the loss of promoters or enhancers or crippling mutations such as frame shift or premature stop codons. Types of pseudogenes. Non-processed pseudogenes. They are derived from gene duplication and are located on the same chromosome as the parental gene from which they are derived. Processed pseudogenes. They arise by retrotransposition and are located on a different chromosome than the parental gene. Then processed pseudogenes. Once again we are going to learn this. Non-processed pseudogenes, they are derived from gene duplication and are located on the same chromosome as the parental gene from which they are derived. Then processed pseudogenes, they arise by retrotransposition and are located on a different chromosome than the parental gene. Then unitary pseudogenes, they are derived from mutations on the parental gene which is in turn lost. Non-processed pseudogenes, they are derived from gene duplication and are located on the same chromosome as the parental gene from which they are derived. Then processed pseudogenes, they arise by retrotransposition and are located on a different chromosome than the parental gene. Then unitary pseudogenes, they are derived from mutations of the parental gene which in turn is lost. So duplicated pseudogenes, original gene is there. Gene duplication, mutations. Pseudogene is produced, retains introns and exon structure, globins and Hox cluster. Here a gene is there, processed pseudogene is produced. So gene undergo transcription, mRNA is produced, processed pseudogene will be created. Splicing, reverse transcription and insertion into the genome. Processed pseudogenes, promoter, transcription, reverse transcription and integration. Unitary pseudogenes, promoter, ancestral gene, mutation and loss of functional gene, promoter. Unprocessed pseudogenes, promoter is there, duplication and mutation, promoter. Then this promoter duplication and mutation will be taking place. First, processed pseudogenes, unprocessed pseudogenes, unitary pseudogenes and polymorphic pseudogenes. So processed pseudogenes, promoter, mRNA transcription, reverse transcription and integration. Then unprocessed pseudogenes, promoter, duplication and mutation. Then unitary pseudogenes, promoter, ancestral gene, mutation and loss of functional gene. Then polymorphic pseudogenes, promoter, reference genome sequence, pseudogene, promoter, non-reference sequence gene. Then pseudogene evolution. Pseudogenes can evolve rapidly because they are not under selective pressure to maintain their functionality. As a result, they often accumulate mutations and deletions more rapidly than functional genes. Pseudogene example, the human genome contained a pseudogene called PSI-GLO that is derived from the functional GLO gene responsible for producing an enzyme involved in vitamin C synthesis. It has accumulated mutations that render it non-functional, indicating that humans are lost the ability to synthesize vitamin C. Role in evolutionary process. They can serve as a reservoir of genetic variation, facilitating the emergence of new genes and functions through gene conversion and exon shuffling.